There we go. Okay. Now, as far as introductions go, normally I would go through and I'd say, oh, our speaker went to this university and got his bachelor's degree, and then he got his PhD, and you get that whole story like that. And I thought I'd try something a little different instead tonight. And instead of describing, his academic credentials, I thought I'd talk about his snake credentials, because he's going to be talking about snakes tonight. And to lead it off, I kind of did a little bit of a survey from Ben's publication list. And it turns out that he has quite an extensive list of publications. The list I found was, so Ben, you'll have to update it a little bit, or I'll give you a chance to update it when you start talking. But this went from 1994 to 2017. So I'm missing the last 3 years. And in that time, Ben has over 30 plus publications to his list on a variety of snakes. And I thought it'd be fun to just kind of list the snakes that he's worked on at some point in his career, with things such as the African brown house snake, Kirtland snake, the short-head garter snake, the brown tree snake from Guam. That's a one that we all should be familiar with. That's the snake that ate all the birds in Guam. The northern water snake, he's got 3 publications on the northern water snake, and he's got 18 publications on the eastern Massasauga. That's obviously has been his favorite animal to go to, was that wonderful snake there. Outside of snakes, he's also done some work on river cooters. Those are turtles. And with that, what I'd like to do is since then, when he moved to St. Louis, he's kind of switched from Massasaugas, he switched to Copperheads. And right now, Ben is at the University of Health and Sciences and Pharmacy in St. Louis. And he's going to talk about tonight the work he's done on Copperheads. So with that, Ben, welcome. And I'm going to shut up and I'm going to give the floor to you.
All right. Well, thanks so much, Rich. That was quite an interesting introduction. You just knocked like 2 minutes off my talk.
Oh gosh,
so
didn't mean to do that.
And do that. So anyway, let's see. I think I do this.
You might want to repeat a couple of them. They're kind of interesting animals.
All right. Did I remember to share my screen?
Let's see if you. I left it on share, so you should have it.
All right. Oh, here we go. That's what I need to do. Let's do this. Oh, did I pick that right? I must not. Oh, I picked Zoom. No. Hang on. New share. Let's pick this one. All right. How about now?
There you go.
Presentation.
Yep. There you go.
All right. Well, yeah. Thanks so much for the introduction. And I didn't know I had that many. I need to go back and count actually, and see what's been happening. No, I'm sure that's right.
You probably have a lot more, because that was only up to 2017.
Well, yeah, but lately, like you said, so I'm originally from, well, here. All right, so we'll just do the whole thing. So, the presentation, I'm happy to chat with everybody tonight. I pretty much have learned to make presentations to keep me on topic. Otherwise, I'll just ramble and tell side stories and tangents, and I'll do my best not to do that. But at any point in time, if anybody has any questions or anything, just feel free to, give me a shout. You don't have to wait till the end to do it. But but anyway, we're going to chat about Copperheads. And I think it's, maybe one of the reasons why I was asked to do this is because this particular project is literally close to home. At the Webster Groves Nature Society. All the data you're going to see tonight came from Powder Valley Conservation Nature Center right there in Kirkwood. I was doing Copperheads a couple years before in Monroe County, Illinois, right across the river, and very southern St. Clair County and very northern Monroe County, Illinois. But all the stuff for this came from Powder Valley, so really close to there.
Hey, Ben.
Yeah.
Quick question before you proceed into your talk, which I can hardly wait for, but I'm just so curious. What is your, what do you do with the University of Health Sciences and Pharmacy? It seems like something different from what we're hearing you're interested in.
Yeah. Well, why do you think that?
Well,
Why
I'm
is
not
that-
sure. I'm not sure what, maybe I don't know anything about that institution.
Yeah, well, you probably don't, because the name of the institution, I'm not trying to be rude, the name of the institution is brand new, but it's been around forever. And we changed our name last fall. We were St. Louis College of Pharmacy, right there off Kings Highway. But then they changed the name of the entire undergrad institution. But I teach in the undergraduate program. So here, let's do a little bit about this. So I'm originally from the St. Louis area. I grew up in Edwardsville. My parents still live in the same house since before I was born. I went to U of I and I started undergraduate research at University of Illinois. That rolled into a master's program where I started with, and actually between my master's program and bachelor's, I was working for the university for a couple of years, but that was all on the Eastern Massasauga rattlesnake. That led to a job in Western Pennsylvania where I was in charge of 6 counties in Western Pennsylvania, the glaciated part of the state where the historic occurrences of the Massasauga occurred. So I was there for 3 years and I was like, well, I like research and I like teaching. I've done both of these things, but I wanted to do it at a higher level. So I wanted to go back and get a terminal degree. And the only person I wanted to work with was back here in St. Louis. So usually at least somebody in the audience knows Bob Aldrich. I don't know if anybody out there knows Bob. He's a St. Louis fixture. He was a SLU professor for his entire career, over 30 years. But I came back and then he started me on water snakes. And we still have a water snake project. One actually that I need to submit within the next few days for the last couple of years worth of research. But
I'm for one that knows Bob.
You know Bob? Oh,
Yep, I know.
Yeah,
him. He's a good guy.
Bob. Yeah, Bob's a wonderful, wonderful person. I love Bob to death. So then I finished up my degree and then I started teaching. My first 6 years, I was at McKendree University. I left there and I was actually, believe it or not, I was headhunted by Urban Chestnut Brewing Company. And I was their, what would they call me? Their senior scientist. They wanted somebody to write grants, analyze data, make presentations and stuff like that. And they liked that I was a home brewer. So anyway, but I did that for a few years. Then I went back basically when my daughter started kindergarten and I wanted I didn't need as flexible of a position anymore. So I went back and started at the St. Louis College of Pharmacy, but in the undergrad. And when I was finishing up my Ph.D. program with Bob, I had heard if you could teach anatomy, you'll always have a job. So I was like, well, that sounds kind of important. Maybe I should do that. So anyway, so basically I teach in the undergrad program. I teach anatomy, I teach physiology, I teach conservation and evolution. But all of my research is always in snake, amphibian and reptile ecology, conservation, etc. But being a teacher is, I can't help but be a teacher. My mom's a teacher. My brother was a teacher. My sister is a teacher. My aunts are teachers. And then I just focused on ecology type things instead of anatomy and physiology. But as you can see, I'm interested in ecology, lots of behavior stuff. Endocrinology was something I started in my PhD program, and that's been really huge in terms of pheromone communication, which leads me to the brown tree snake and things like that you were chatting about, Rich. And then I've worked on a number of things, starting with an endangered salamander, then an endangered turtle, then an endangered rattlesnake, and now 2 things that are not endangered. Although the copperhead, is one of those things that we just don't have a lot of good data on. And I think nobody really has a good handle on what population sizes are and if they're really declining or not over a period of time. But yeah, St. Louis-ish native. But yeah, Anyway, all right. So I think a lot of people think about snakes and they think of something like this. This is a northern water snake. And to me, this is how I will always picture water snakes, is they're foul and intemperate little beasts looking to bite. One thing interesting, does anybody know what this, I don't know if you can see my mouse on your screen, what this is here. All snakes have this. Here's the tongue, where the tongue would come out. So it's not a hole for the tongue. Anybody know what this might be? I'll give you a second to ponder what you think it might be.
Is that so they can breathe while they have their mouth full of fish?
Yeah,
something?
you got it. You got it. I mean, you know, snakes, they have a very flexible jaw. They have cartilage between their mandibles here, so they can expand their jaw, and they can swallow something so big. In fact, we had 2 instances of a snake that ate a meal so large that it asphyxiated itself. His lung couldn't inflate. The meal was so large that it basically died from asphyxiation. But that's the trachea, and like our trachea is lined with cartilage, but in case they have their whole mouth engorged and no air could pass through, right? This is how they could always get air down to the lungs. But anyway, this is how I always picture water snakes. And when I describe what I do for a living, I saw this at the St. Louis Art Museum one of the last times I was there, and I was like, oh, this is perfect. I imagine this is how people see my job. In fact, everywhere, they're like, oh, and at my daughter's school, her Girl Scout troop, everybody in her class knows me as the snake guy. And I'm like, I do other things. It's not just that, you know. But anyway, I picture when people think of what I do and go out and you know, spend a considerable amount of time out in the field catching snakes, studying snakes, that certainly something like this must cross their mind.
But do you do it naked?
Not with copperheads. It's they're venomous, you know. But the water snakes, yeah. I mean, you're in and out of water. Why not? So, yeah, good point. Good question. All right. Usually people finish presentations with acknowledgements. I like to start because none of this would happen without all the help that I have. I drag my daughter out constantly. She just thinks all 8-year-olds do this. She doesn't realize that she's being forced into child labor. I've had a lot of student volunteers over the years, volunteers from Powder Valley that are out there. The zoo has been one of the biggest partners since 2000. I've worked with the zoo, St. Louis Zoo, on a number of different projects, but they provide all the veterinary care for the snakes. They do all the radio implants. Anytime a snake is unhealthy, you know, we just go and they check them out and give them whatever meds they need. Powder Valley itself, lots of great help at Powder Valley. And then even with educators at the St. Louis Zoo, like Brittany, who would come out and volunteer. So again, lots of help over the years, and it wouldn't keep going without everybody. Also, no research is done in a vacuum. Here are a lot of the partners that I have: the Natural History Survey up at U of I, the St. Louis Zoo, of course, my institution, who's, you know, for some reason thinks it's cool that I do this, and sometimes will give me a little bit of money to do it. SLU, because I still work with Bob quite a bit. Missouri Department of Conservation, of course. So lots of collaborators that help make all this possible. What I thought I'd talk about tonight, just a little bit about the ecology of copperheads in particular. I titled this In a Fragmented Landscape because, as we'll see first in a second, when we look at a map of Powder Valley, really how isolated it is. It's really just one little island of habitat surrounded by interstates and the Merrimack and then suburbia Kirkwood. And then we're still collecting data. I don't have any of the 2021 data entered, but we do have 2018, 19, and 20. So the preliminary results I'll show you, it's certainly preliminary, but it's based over 3 years worth of data. So it's getting close to probably figuring out what they're actually doing. So we'll talk a little bit about their spatial ecology, including habitat use, overwintering. And then one thing that's really been surprising to me is simply the mortality rate at Powder Valley. And then some other things that we found along the way and some things we're still working on. So that's kind of the scope of what we'll do. So we'll go through the introduction and the project origin. Basically, this was a project that was supposed to be done by the St. Louis Zoo, but then the curator, Jeff Etling, left to take a position at it's either Wichita or Topeka Zoo, where he became the director of the entire zoo. So they didn't have anybody to do it. And I was moving over to Missouri, and I called Jeff Brigler, who's the state herpetologist, to get my permits transferred over. And he's like, it's like, boy, I have a project for you. We'll buy you all the transmitters if you have the time to do it. I was like, okay, that sounds great. And it turned out to be the Copperheads. And it was at Powder Valley. And I was like, I just moved to Kirkwood. I could ride my bike there. And literally, I've ridden my bike there over 100 times. it's exactly 3.5 miles from my doorstep to get to Powder Valley. So, but anyway, so it started in 2018. I chatted with what the state wanted, then of course what Powder Valley wanted to get out of it, what MDC and Powder Valley itself wanted to get out. And they basically wanted to know all about the ecology, and they wanted to then incorporate what we learned from the ecology of the snake, particularly at Powder Valley, into public education and public perception. It has annually 80,000 visitors. The trails are paved. And so the opportunity for human snake interaction is ripe. And there have been recorded bites of visitors from Copperheads at Powder Valley. And basically none of those have happened since the project started, but 2 of them happened within the last 10 years. And both of them involved Copperheads, of course. And both of them involved kids that were wearing flip-flops that saw a snake on the trail and tried to shoo it off the trail with their foot, and the snake bit them. And that's essentially how it happened. So the goal was to learn as much about the Copperheads, if there's spots where they cross the trail more or less often, times a year they're most active, to then relate that to the public, and also for any habitat management that they may want to do at Powder Valley. So over the years, we just eclipsed 2,000 total radio locations. Those 2,000 radio locations, we've found 33 individuals, which is roughly an average of about 6 individuals per year, because some of those individuals encountered were babies that were born to an adult. So it was kind of pseudo replication, if you will, in your data set, a way to build up numbers really quickly. But we found about 24 individuals roaming around over the past, like I said, 4 years. So average about 6 years or 6 individuals per year. Not great if you're really trying to build up a data set. It takes a long time if you have that small amount. Of those 33, we put radios on 25. 16 of the 25 were adults or subadult snakes, and then 9 were neonates, including 6 individuals from 1 litter, and then an additional 3 that we found this past year. Snakes have been on the air anywhere from over 3 activity seasons up to 304 radio locations. So one single snake relocated 304 times. Over 304 days where that snake was. And the shortest one before it basically got eaten was 8 days. This year was really hard on the population. We lost quite a few snakes, and right now over winter, right now we only have 2 snakes remaining on the air. We started off this season with 8, and lots of mortality has happened this year. So a little bit about the Copperhead itself. We'll just focus on Missouri. I taught evolution, and this is somebody's own personal appearance. If you want to chat, or personal preference, if you want to chat later, we can, about what your thoughts are. But if you believe in subspecies, here's the range showing all the different subspecies of Copperheads throughout the United States. If you don't believe in that, that's fine. I don't know if I believe in it anymore. But you see the map, the whole map is where Copperheads occur, all the way northeastern, like New Hampshire, right, and all the way, but down a little bit into Mexico, and you got the Trans-Pecos down here in Texas. Where Missouri is, particularly St. Louis, I put this nice little dot on the map. You all know that we're here, but there's 3 different intergrades of populations that all occur right around the St. Louis area. So one thing that we do is we take genetic information from all the individuals. I think looking at the population genetics of these particular individuals might be pretty interesting in an area where you could have intergrades between these different subspecies. But you look at you know, you read all the literature, you look at the natural history of these things. They like oak hickory forests, particularly with hillsides, rock bluffs, and you typically hear of adjacent open fields, and that's basically meadows, what have you, for foraging. And if you've been to Powder Valley, it kind of has all of that. And they're just creating, as of fall 2020, so it's been just over a year now. They started right behind the administration building. They cleared a little bit of area and they're planting it in prairie. The typical, besides that, the only really open areas they have are the grassy strips along the entrance drive and right around their parking lot for the employees. There's a garage and has a little bit of gamma grass, big blue stem, and some other prairie plants in there. And you'd be surprised, but copperheads flock to those areas. There's the little bit of open grassy areas that they have. And we're starting a project with rodent trapping and looking at rodent densities in those grassy areas versus out in the more wooded areas, and see what the rodent density is between there. So Powder Valley fits that pretty well, except the open areas, open grassy areas aren't very robust. Pretty much they'll hibernate. You know, you get down in Louisiana, they may not need to hibernate at all. Probably is not getting really deep down in really cold temperatures, but certainly up in the Northeast and here in Missouri, if they don't find a place to stay above freezing over the winter, you know, they'll turn into little snakesicles. So pretty much they'll hibernate anywhere that they can stay at least 4 degrees C, whatever that is, 38, 39, 40 degrees Fahrenheit over the winter. But you hear a lot of them hibernating on southwest or sometimes even north facing slopes. They've been known to hibernate individually and in groups, so colonial or single hibernators, in caves, gravel banks, animal burrows, particularly small rodents, hollow logs, sawdust piles, cracks of building foundations, etc. In fact, Bob Aldridge, I just got another email from him. His son, one of his sons, lives near here and has had 4 copperheads make it down into his crawl space looking to hibernate this past year. Just this past winter. I'm not sure exactly whereabouts his son is, but like we could use that in our data set. And one thing that's also interesting that's popped up is really copperheads at Powder Valley are middle of the food web. People think of them, they're definitely not apex predators. They get eaten by pretty much everything. Other snakes, king snakes, racers particularly will eat them. Possums will eat them, raccoons, coyotes, foxes, feral cats, turkeys have been known to eat baby copperheads, birds of prey, of course, and even moles. Moles have been noted, I think it was by Henry Fitch in Kansas, to tunnel up underneath a baby copperhead and then pull it down into its burrow, basically snap it in half and eat the babies. So, like I said, we really don't have a good handle on how robust the populations are. But I found this quote and thought it was kind of interesting, where it says, because of its secretive nature, and in a second you'll see some photos of why copperheads are so hard to find, but because of its secretive nature, the copperhead often survives in relatively close proximity to large human centers, such as Powder Valley. And as urbanization increases, however, it tends to disappear except in those protected areas that also provide an abundance of hiding places. But the problem we have here and nationwide and even globally is, of course, those protected areas are getting smaller and smaller and smaller, and humans keep encroaching on habitat. So what the population numbers are, you can ask anybody and no one will give you a direct answer, but they'll all say, I don't think there's as many as there used to be. And you hear that a lot for a lot of different species. Powder Valley itself, 112 acres of this oak hickory habitat. As you can see here, you've got Interstate 44, southeast side, Interstate 270, southwest to west side. You got the Merrimack River, not too far by. And then everything else is suburbia. And these snakes do not like to leave Powder Valley habitat. And I can give you an example. I had this one snake this year who was found hibernating off-site. We'll chat about that in a little bit. And then went all the way to the very edge of the habitat where it was suburbia, turned around, and within 2 days went all the way back across where Craigwell Road is here. Hit that road, turned around, went up to where it ends, this is private property here, hit their driveway, and then moved back into Powder Valley. Like, it explored pretty much every possible opportunity to escape. It went and hit the open canopy with no cover, no protection and roads. It immediately turned back around and went into the Powder Valley area and habitat. But really, this is this isolated little patch of habitat. There is Emmenegger not far away, and there are copperheads there. I have one copperhead cross Craigwell Road. I found a couple that were hit on the road on the way to Emmenegger, but I've not had any copperheads that had radio transmitters leave Powder Valley, make it over that little I don't know what that little road that goes over the interstate and into Emmenegger. So I don't know if there's any migration between those two or not, but it's certainly its own little patch of habitat. Well, if you want to do a study on snakes, you want to do anything about movement, growth, hormone levels, you have to find a snake, and you have to find that exact same snake again so you can measure it again or look where it is and see what's different between time 1 and time 2. And that would be great if they all looked like this. It'd be really easy to find them. Like, there's no problem. This is about 1 in 1,000. Usually what you see is something like this. So use your best eyeballs and see there is a copperhead visible in this figure. So we'll focus on the one on the left and see if anybody can spot it. Whoever spots it first gets a taco that Richard's buying, he said.
I better find it first then.
It's smack in the middle there. I think I see a little bit of its pattern over the.
All right, tell me if you can see my mouse. We'll play warmer, colder.
I see its head somewhere, I think, on the left side.
Left
All right, how am I
side.
doing?
Closer.
Left.
That's it right there.
Here. Yeah,
That's
you
it. You're
can
right on
also-
top of it. That's it.
Yeah, right here. Not all that easy to spot. It's still pretty open. You can see how their camouflage is. What about this picture? There's a copperhead somewhere in here too. So,
I definitely saw that faster.
you saw that one faster? All right.
Middle, little less than lower middle. Keep going
So
down,
here.
a little further down. Go back up. Go back up. Almost. Keep going a little bit. There you
There it
are,
is.
right there.
Yeah. That's usually all you see is usually a little chunk, if that, but they're buried underneath the leaves. I took a picture this year of just one with its head. All it was his head sticking above all those oak leaves. And I got down on the ground and took a picture of it, looking right at it. And I was like, this is probably the last thing a mouse ever sees. It's running along and then just this head up and then right there. So anyway, they're extreme.
I just threw him out because he was bothering the cats and I was trying to listen to the talk.
I'm sorry, was that directed to me? I didn't hear that.
I think James took off with some event going on. We need.
Oh,
Yeah, Jan's home. She's home.
all right. So anyway, yeah, they're really difficult to find. So if you want to find them, very highly camouflaged.
Oh, he's better.
They can move over 100 meters in a building easily. Without
You have the power to mute, Rich.
without something to even the playing field, these types of studies wouldn't happen. So what do we do if we're lucky enough to find one? These are actually old photos from McKendree. But we'll take the snake, we'll measure it, we'll weigh it. A lot of times we'll weigh it in the field with a very official pillowcase. You measure the pillowcase plus the snake, and you subtract the pillowcase off, and there you go. All the snakes are individually marked with a pit tag, same thing your dog or cat gets at the vet. And then we take blood sample can be used for paternity analysis, which we're doing for overall population genetics, and for hormones. Like I said, I'm really interested in snake pheromones over the past 10 years or so. And then a subset of individuals that are big enough get radio transmitters. So the zoo's we're lucky enough to partner with the zoo on this. And this is really eye-opening if you ever get the chance to see a procedure like this. But they basically anesthetize the snake, and then they'll go through and they'll use this basically like tongue depressor to hold the fangs. The fangs are retractable, so they're on little hinges. So they put the fangs, tuck them up underneath, and they tape the tongue depressor around the top of the head of the snake. So even if the snake wakes up during surgery, it can't bite anybody. They will stick a little catheter tube down the trachea, and then they can control the oxygen and the anesthesia that the snake is doing, so they can wake it up when they want to, knock it deeper down into sleep, if you will, over the procedure. And then the procedure itself is really simple. They just make an incision about 2/3 the way down the body. They cut the skin, they cut the muscle, and then the snake's body is basically just a whole bunch of ribs. So they just spread the ribs, and then they get the transmitter. You can see here, transmitters come in different sizes. We have to do it within 6% of the snake's body mass, no more than that. Then the transmitter gets inserted into the peritoneal cavity. So it's not in the stomach or in the intestines, but next to it, and just kind of squishes all that off to the side. And then the antenna, the transmitter itself has an antenna. You can kind of see it in this lower left picture. They run the antenna up subcutaneous as opposed to through the peritoneal cavity. That way it's closer to the surface of the skin, and it's usually straightened out, so we get a better, clearer signal from further away. Then they give the snake some fluids, some antibiotics, a little bit of painkiller, and they wake it up and say, okay, hang on to it for 24 to 48 hours, and then we let it go exactly where we found it. No snakes are ever moved. If you ever hear somebody saying, oh, they dropped these snakes from black helicopters and blah, it's not us, not us. We never had anything to do with anything like that. In fact, nobody I know has a budget to be using black helicopters to be dropping snakes anywhere. So anyway, so we put the transmitters in, we let them go, and then we track them. We track them every day, 7 days a week. We'd always make jokes in grad school that snakes didn't know it's your birthday, snakes didn't know it's your girlfriend's birthday or your Mother's Day or whatever. 7 days a week from the active season, which is basically late March to early April through the end of October. The transmitters that we use are temperature sensitive. So for example, the warmer it is, the faster the transmitter will beep. And the transmitter manufacturer company will send a calibration curve where you can then determine by how quickly the transmitter is beeping what the temperature of the snake is. And that's not all that interesting throughout the regular active season, but when they get down into hibernation and you can't measure them with an IR gun or measure the ground and the air temperature, kind of assume what they're going to be, then it gets pretty interesting. So we've been able to figure out at what temperature these snakes are hibernating at over the past couple winters, and I'll share some of that with you in a little bit. So that's what this is. We just go and we take the pulse rate 3 times a week over wintering to get a good thermal profile of the snakes while they're hibernating. When we collect anything about the encounter, the snakes themselves, the habitat, morphological data, genealogical data, anything that we can collect, basically, we're collecting. In terms of how we organize it all, we use the data program FileMaker Pro. A lot of times I do stats either in SPSS or Excel. We use Google Earth as well. I'm not crunching any numbers for you all tonight. Like I said, it's 3 years worth of data. I have some averages, but I'm not looking to see you won't see any p-values or t-stats or chi-squared or anything if that's what you're looking for. A little bit about what we found so far. So this is one snake. This snake was on the air the longest. He was actually the first snake that was found. We had to give him a good name. And we figured, well, it's, Kirkwood. So we named him Kirk and then thought that wasn't cool enough. So we named him Captain Kirk. And everybody loved Captain Kirk. Everybody would ask about Captain Kirk. We found him right here crossing, what was that, Tanglevine. And then we found him in September. So this is the same snake, Captain Kirk, over 3 different years, from September till he went into hibernation the first year. Here's the second year, 2019, he was on the air the entire year. And then the third year, he came out of hibernation and he lasted till late July, and then he got eaten But you can see the time of year really makes a difference in terms of activity range size and what all they're using. So this particular snake, September to November, basically found him here. He didn't do much, and he went. And this is one of the more interesting things: is he hibernated in the riprap underneath the shoulder that built up Interstate 270. Not only did he hibernate there that year, he hibernated there the exact next year in the exact same hole in that riprap. So he hibernated there 2 years in a row. But look at the difference in size. When we have him on the air, right, collecting data from the entire activity season, we get an activity range size of 17.4 hectares, basically double what he was just in the fall. And you look at this last one, 2020, that was 2020, when the poor guy got eaten. Activity range size, I don't know what it is hiding behind my view here. 3.3 hectares. Right? What this one here does not include 2020, what 2019 does not include is the mating season. And look how much more the snake moved during the mating season. This has been consistent no matter what snake species I've studied, is males move in search of females. Now they're not doing this blindly, they're looking for female pheromone trails. And how they find those pheromone trails is a big part of a different research project that I'm doing. But also it really ties into this as well, is during the mating season, this snake basically went all this entire distance within 10 days until he found a female, and then he stopped moving. So really activity range size can vary drastically throughout the year and is positively and directly linked with the individual snake, whether it's male, whether it's female, what time of year it is, and what the snake has on its mind. Another time we'll get really big movements is right before hibernation. There was one snake that hibernated underneath Craigwald Road. I don't think any of these, this could show it decently enough. And it was the snake was found right by the administration building. So let's just say here, right by the parking lot. And within 1 day moved about 250 meters to Craigwald Road overnight, just one night, because it got cold and he didn't want to get caught above ground and freeze. So they went straight into hibernation, and that was it. We didn't see him hardly at all the rest of the year. So they can move a considerable distance when they want to. Here's a couple others that also show the same thing. Here's a male. We found him right before hibernation. Relatively small activity range size, 8 hectares, but you get him the entire year. The next year, 22.1 hectares. And again, it's a male, and the difference is this activity range size on the right. It encompasses the mating season. The snakes, male snakes, are looking all over for lady snakes. A little bit about one of the things that Powder Valley particularly wanted to know with their 80,000 annual visitors. Distance to trail, like how
Ben?
close are they? Yeah.
Ben,
Yeah.
Sorry to interrupt on this slide. Back on the previous slide, we had a question about
Oh, sure.
so the snakes that were eaten, do the transmitters survive being eaten, and are they left with the carcass of the snake or the scat of the animal, or scattered someplace else in the forest?
Great question. The answer is all the above. One time I had a snake was in this one particular spot. This wasn't this particular study, but this one spot, go out the next day, no signal. Like, okay, this happens because snakes can move a considerable distance, sometimes out of the range of your receiver. So at that point, you start hiking. You hope you bump into a trail. If that doesn't work, then you get in your car and you drive around and hope you bump into the signal. And none of that worked. We looked for the snake for over 3 hours one day, drove everywhere, hiked every trail possible, nothing. So then the next day we come back and like, okay, let's just try it again. Turn it on, amazing signal. Takes us right back to where we were 2 days before, the place where we had the last signal, and we found the transmitter in a pile of fox scat. So the fox ate it, ran off somewhere, came right back, and apparently crapped it out right exactly where he ate the snake, I guess. But anyway, you can usually tell what happens, what the snake was predated by the state of the transmitter. Most of them at Powder Valley have been raptors. And the reason that we can deduce that is you usually find the transmitter right underneath an old dead branch, usually right over a nice spot in a field overlooking a field. And you find the transmitter just in one little chunk of body. I don't know if you can see me. You might just see my screen. I'm trying to motion with my hands. About the chunk, about the size of the transmitter. And it's sheared off nice and neat on either edge. And it's just the chunk of the body that has the transmitter in it. And that's almost always from a raptor, with those really sharp edges on their beaks to cut. And the transmitter just drops down, and then they eat the rest of the snake. If it's mammals, coyotes, foxes, raccoons, believe it or not, what have you. A lot of times you'll find the transmitter chewed on. You'll find little tiny teeth marks in the transmitter. The antenna wire itself will be frayed and chewed on. I guess that really, because of the smell, really tastes and smells like, part of the body. And then they bite it and like, oh, I don't want this, and spit it out. So it really depends. But you usually don't have a problem recovering the transmitter. But usually it needs to be refurbished before you use it again. So hopefully that answers your question. And if not, let me know. I can elaborate if you need.
How long does the battery last?
Well, it depends. So the bigger the transmitter, the longest we have goes about 18 months, and that transmitter weighs about 10 grams. Some of the other ones we have, the transmitter weighs 3.5 grams, but only goes 8 months. And then for the babies, we use a transmitter that weighed 0.9 grams, but it only lasted 60 days. So the battery makes up the bulk of the weight of the transmitter, and the bulk of the weight and the bulk of the size of the transmitter. And then the weight of the transmitter is what governs, the size of the snake that we can put it on. So obviously, if you find a really big snake, that's great, because you can put a transmitter. And again, these snakes have to go through a surgery, right? They have to go through, cut through muscle, spread ribs, cut through skin. Everything has to heal. It's a pretty invasive procedure. So if you can avoid doing that by putting a transmitter with a battery that lasts longer, that's great for the snake. But you also can't have something heavy. It's like you carrying around a bowling ball all the time. You can't have it be too heavy and really impact any part of their natural history, where it would prevent them from escaping from predators or be in the way if they need to develop and then birth offspring. So a lot of consideration has to go into it. But the rule of thumb is, 6% or less of the snake's body mass is what our IACOC, institutional animal care and use committees, allow to happen. So that's pretty much what determines it.
And remind me again, are copperheads live bearers or egg layers?
Yeah, so in North America, the pit vipers, so your rattlesnakes, copperheads, and cottonmouths, and then your natricine snakes, which are the water snakes, the garter snakes, queen snake, line snake. All those are live bearers, or what they call ovoviviparous, where they're born basically live, definitely alive, but without an egg, but encased in a membrane, as opposed to racers, ringneck snakes, every other snake species besides the ones I mentioned, rat snakes that would all lay eggs, like a leathery egg. But yeah, the rattlesnakes, the Echistrodon, the natricines, those are all live bearers.
So what I'm getting at is, okay, would that transmitter get in the way of if you put a transmitter into a female snake, would that get in the way of her laying eggs and or live offspring?
It shouldn't. However, if we find a female early in the season and we have radiographs and ultrasounds done on every female to see, one, if we can get an estimate of litter size. You can usually count and palpate and feel the eggs developing ova inside, and get an idea of what it's going to be. And if it's early in the year, I have no problem putting and neither do the vets have a problem putting a transmitter in a female that's going to give birth that year. If it's later in the season, within a month or so of giving birth, we never do. We wait till after she gives birth and then put a transmitter in her, so we'll hold her in captivity. And again, it's a really invasive procedure, and I'll get to the baby snakes and what we did. But I didn't want to put them through a really invasive procedure right before birth. But early in the year, it's okay. They recover fine. They have enough resources. Think of all those resources that are going to those developing offspring. And if you have to use resources, energy that you're, eating and bringing into your body to use for healing, then you don't have enough as much for your babies. So we try to be very conscientious of that. Okay, can I go on? Are
Thank
we
you.
good?
Sure.
Sure. Okay. So anyway, Powder Valley is interested, distance to trail, distance to road. We're looking at forest edge, canopy cover. And, the thing about statistics is, there's lies, damn lies, and statistics. So this is one of those things you start looking at, and it's like, oh, all right, well, look, distance to the nearest trail. 21 meters, 30 meters, 22 meters. They're not hanging out too close to the trail. These snakes that we have transmitters in, they're not really hanging out, except for these couple. They're not really hanging out too close to a road, 30 meters away, right? 100 feet away from the road. We shouldn't be too worried about that. But when you go into the fine scale and start looking at this, you start to see a different pattern. So this first snake, I pretty much use Captain Kirk for everything because we had 309, 340, whatever it was, radio locations. So we have the most data on him. So we'll look at that, and here he is, snake one. Even though on average, right, he was located 22 meters away from a trail. Here's him, and here's a trail, right? So I'm walking really close by. So I made an arbitrary cutoff of within 5 meters of the trail. And then I started looking, and within 285 locations, he was located within 31 meters or 35 meters of the trail 31 times, or 11% of the active season he was there. Including 9 consecutive days. He was encountered 0.2 meters from the trail the day before Independence Day. And I only mention that because I can only imagine how many people will be walking the trails on Independence Day. Didn't want anything bad to happen to the snake or to people. And he crossed the trail over those 3 years at least 10 times that we know of. So even though it seems like they're far away, they go back and forth. They're found near the trails. A lot of times near the trails are edges, and where the edges are, it's a boundary that they don't want to cross, but it's also a good food source. And with the same thing for the road. So if you just look at distance to road, average about 30 meters away from the road, no big deal. But if you start breaking down the data and look specifically, about 20% of the time he was found within 5 meters of the road. It's just there were some other days that he was really far away from the road to balance that out and make it look like he was, 30 meters away from the road on average. So he was within 5 meters of the road for over a month at a time, right? 35 consecutive days, over 10 different sites along that road. Just kept moving up and down the shoulder. I think it was that entrance drive, and my guess is he was looking for food, just kept foraging. We have snakes a lot of times move out to that grassy strip near the drive, the entrance drive, and then move back into the woods just to move back out to that grassy strip a month later. So my guess is they're foraging, using it a lot for foraging. He overwintered underneath Interstate 270, the two years that we had him. And he crossed the entrance drive at least twice successfully. Wasn't hit by a car, wound up being depredated. Other examples, snake number 5, encountered near a trail, again, 10% of the time. Another snake, 20% of the time. And you can see near the road, right? So a lot of those stats, you start looking at averages that can be a little bit deceiving. So the question is, why? Right? Why these stretches where they're found near the road, near the trail? And again, to then use that in public education, public programs, and also to inform Powder Valley, maybe put up some signage. Do not shoo this snake off with a bare foot, type of thing, because it could be. But again, they're so cryptic, so camouflaged. A lot of times visitors have no idea. And in fact, I was walking behind some people one day, and they stepped over Copperhead and thought it was a stick, and just kept going. So I think it happens. People come in very close contact, and I'm out there, like I said, 7 days a week, and I love Powder Valley. And if you ever go to the Nature Center, it has all those activities for kids, and I love that kids are out there and hiking. And I cringe though, because the parents let their kids go off and pick up sticks, pick up rocks. And I tell every one of them, like, it's great that you're exploring, but there are things out here that could hurt you, including venomous snakes. And instantly like, oh my God, and they pick their kid up and then like, stay on the trail, type of thing, which I get it. I have my daughter go out. She's, like I said, she doesn't really know any different. She goes out, but we have very specific rules about where she can put her hands and not put her hands. And she's gotten to be quite the naturalist over the last few years. But it's pretty interesting how close they are. And I bet how close in contact people come to them and just don't realize. So we also did the same thing with baby snakes. So one of the snakes we had gave birth in captivity. She ate birth one morning before my daughter went to school. She was eating a Pop-Tart. And I was just like, Dad, look. And she's like, this is gross and cool at the same time. And I was like, that's probably the best description that anybody's ever come up with. So we had a litter of 6 babies. So we had a transmitter on mom and then a transmitters on 6 babies. As you can see, we did not put them through that procedure. The transmitters were externally attached instead of the surgery. And again, my thought was, you're born, you need all your resources to go into growing. We don't want to have to make you heal this big cut, like, hey, welcome to Earth, right? And cut them open and stick a transmitter in them. That didn't seem like the way to greet anybody. So we glued the transmitters on them and monitored the baby's movements in relation to mom and just in relation to what they did, because nobody really knows what neonatal snakes do. One thing you notice is venomous snakes, as babies, they have this bright yellow tip tail. You see it here, you see it over here. As for caudal luring, so they lure their tail back and forth. It looks like a worm or something that, some little rodent would be interested in eating, and they come up to, grab the tail and then realize that it's not exactly what they thought it was. So I can give you a little bit of preliminary data on the baby. Oh, and this is how they're born. So when they're born, I said they're born live, but encased still in a membrane. You can see the little sheen here. They're still in this, you can see little blood vessels down here. They're still in this membranous sac when they're born. And again, it's not true viviparity, which literally translates to live birth like humans, but ovovivipare. So it's like the eggs are retained and you just get this little membrane. And really, it's kind of interesting. The snakes are born live, but they have to get out of this membrane within the first, seems to me, 5 to 10 minutes, or else they run out of oxygen. Not all the babies actually poke their heads out of this little membrane for whatever reason. You can speculate all you want, natural selection and evolution. I don't know the cause. But here's the size of when they're born. They're basically, and the size of them, the best thing I can say, they're like a little number 2 pencil. It's about the length of them and the width of them. That's basically about the size. They can curl up less than a quarter. So as tough as it was to find the adults, good luck finding these little babies out in the wild. So what we did for the babies, again, they did not go through a procedure, but I invented the invasive surgical procedure, but the zoo and I came up with a new way to do this. We're going to write it in a little paper where basically we glued the transmitter on with surgical adhesive, but that doesn't always work because literally because the glue manufacturer changed their compound that they make, the formula, and it no longer sticks very well to snake scales. And I didn't want to lose the battery. So we decided to suture the transmitter, just run a suture underneath the skin, but then tie the transmitter onto the body of the snake. And that seemed to work well. We had no problem. Maybe 1 or 2 of the 9 transmitters popped off, but the rest worked really, really, really well. So what did we find when we did this? Well, basically the neonates are exactly like miniature adults. And this particular litter is, I think, more subject to the particular habitat it was born in. But the one thing to keep in mind, and I didn't really realize this, is I haven't kept up with literature in all the fields as best as I could have. But my master's advisor at U of I, his name was Chris Phillips. He's an interesting person. But I was chatting with him over the fall, and he goes, well, yes, it's the 20% rule. He said, basically out of all the animals studied, 1/5 of them venture off. The other 4 stayed close to home. And 1 in 5 typically ventures off, moves away, dispersal, genetics, to limit inbreeding, what have you. And you can see this, right? You have one snake here that does not really look like any of the others. Canopy cover, 72, 73% versus nobody else above 16%. Distance to porous edge, I haven't calculated yet, but it's a considerable distance, much more than this. And this snake, this bottom one, and again, think this is something the size of a pencil. And think of the terrain, hills, rocks, logs. This baby snake, the size of a pencil, in one day moved 106 meters. So more than the length of a football field in one day, over rocks, logs, over a trail, did not get eaten. And I literally thought because I got the signals coming so faint. I thought, oh my God, something, ate it, dragged it off. I'm just going to find the transmitter. And nope, I lifted up a little rock and they poked his little head up like, what are you doing? Who turned on the lights? But it was pretty amazing. We didn't have the best luck. 2 of them got hit on the road trying to cross the entrance drive. One of them was eaten. One of them died of natural causes. And of the litter of 6, 2 lived until their first hibernation. So again, mortality is really, really rough on snakes of all ages out of Powder Valley. There's a little bit of habitat. The adults seem to be found in leaves and rocks primarily. In terms of retreats, they use a lot of rocks and logs. That seems to be the basis of it. And again, the neonates, this was one litter, and so I think it's not really indicative of what the snakes would have been doing if they were born someplace else. This particular litter was born in that grassy strip right along the roadside. So of course, then vegetation was the predominant substrate that they were found in, as opposed to rocks or leaves. But retreat was the same. No matter where they are, they'll try to find rocks and logs and get away from predators and climatic variables as best they can that way. Activity range size, here are those 2 neonate. Here's the one that moved basically 106 meters within one day, straight out in the middle of the woods, found a creek bed, and then it hibernated near the creek bed. All the other ones never left this roadside. You can see here's the entrance drive here. This is the grassy strip on one side. This is shade over the grassy strip on the other side. But the other 5 did something similar to this, where they had 0.004 hectares was their size. They basically did not move much. And we found that with other snakes too. I put transmitters on neonate Massasaugas. They dispersed a little bit and all came back to the same rodent burrow where they were going to be born to hibernate. So they don't all move very far.
I got a quick question. What happens when those neonates need to shed?
That's a good question too. So I waited, they'll shed usually within the first 10 days after they're born. So I waited till after they shed to put the transmitters on. But they've had no problem shedding around the transmitter. It just breaks off the front half of the shed, breaks off. The part with the transmitter's still stuck on it a little bit, then the bottom half comes off too. So they basically shed around it. But I waited till they had their first shed to avoid it as much as possible. And let's chat a little bit about overwintering. So one of the things that really struck me was the first year we had 3 snakes. The year that the study started, we had 3 snakes that all went into hibernation. 2 of those 3 left Powder Valley completely to hibernate. One went underneath Craigwald Road, which is the road that goes to the entrance drive of Powder Valley. And then of course, I mentioned the one that went underneath Interstate 270 to hibernate. And since then, that has held true. Of the 11 individuals that have made it through hibernation, and 11 different individuals that have made it to hibernation in multiple in the different years that we've been doing the study, almost twice as many go off of Powder Valley, even though it's 112 acres of great habitat, to hibernate. Only 4, half that many have stayed on grounds to hibernate. Where they hibernated, typically rock embankments along the entrance drive, 2 along Craigwell Road, and then 1 for 2 years underneath the riprap in 270. Soil and rock embankments, a lot of them off-site, 4 of them off-site, 1 along the entrance drive, and 1 in a rodent burrow or root system of a tree. But they're finding these hillside slopes, and this is a lot of man-made habitat through natural habitat where the ground was already rocky. But we may have accidentally, and this is a happy thing, we may have accidentally created some habitat when they built Interstate 270, particularly for snakes to hibernate under. And I mentioned that the radio transmitters are temperature sensitive. So here's their temperature profiles for this. These aren't average. This is the average for the month for each particular snake. So as you can see, the coldest month is typically February, right? All around 10 degrees in November. December, a little bit colder. January, colder yet. February, down to 5 degrees, usually between 5 and 8 degrees seem to be what they're hibernating at. Minimum temperature any of these snakes reach throughout hibernation, again, 4 degrees C. So I'm thinking high 30s, low 40s, somewhere in there is typically what they're keeping their body temperatures at. And to me, how do you, and I was asked this just the other day, how do snakes know when to come back up? I'm like, I have no idea. Think about this. You go down in a hole in November, maybe late October, no sunshine. You don't have an alarm clock or calendar down there. And you're just down in this dark hole for December, January, February, March, 4 or 5 months. And then all of a sudden, you know when to come back up. So I really think it's the temperature of the ground. When I started looking at this data for these few snakes here and noticing their body temperatures when they started coming up out of hibernation, it was basically when their body temperature reached somewhere around 11 degrees for about 3 or 4 days in a row. Then they come out of hibernation, but they stay right next to their burrow. Then they go back down at night, come back up, go back down at night for a good another 3 or 4 weeks until they feel, I think, well, this is my guess, until they feel safe enough to move further away from their burrows. But to me, it's got to be a temperature. And again, the ground is much more stable than fluctuations in the air. So they don't know that it's sunshine. They don't know the particular day is 70 degrees and they should be up. They have to wait till the ground temperature 2 or 3 feet deep gets warm enough and warm enough consistently that then they feel, they can come back out without really risking mortality rates. Mortality rates has been huge. This poor snake drowned. She got caught in a flash flood. As you can see, she's 2.5 feet long. This is typically a dry creek, except during flash floods, and snakes 2.5, 3 foot long. Her body got swept onto this root that went across the stream, and then water, and she couldn't pull off it because of the force of the water going down. Leaves, sticks built up behind her, and she just was hung up there. Found her the next day. Took her to the zoo for a necropsy, and they concluded that she had drowned. She had also eaten a mole, and she had 3 babies that she was going to give birth to. But one thing that's striking about this study is the mortality rate. Of the 16 radio-equipped individuals, 5 have been eaten. 3 transmitters have died prematurely. 1 just happened about a week ago. Again, 2 currently on the air. 2 had complications from surgery, so we took their transmitters out and told them they graduated. See you later. 1 was hit on the road for the adults. We had 2 babies hit on the road, but 1 adult hit on the road. DR is dead on road. All these acronyms are different. My aunts were nurses, and when I started talking about the DNR, for Department of Natural Resources, they kept wondering what do not resuscitate had to do with anything that I was doing. But DOR, in case you don't know, is dead on road. 1 drowned, right? That's this one here. 1 asphyxiated in a burrow. It got trapped between a root system and a rock that it literally could not come out of, and then died overnight. And 1 has died during the overwintering. Of the babies, I showed you those first 6. 1 was depredated, 2 hit on the road, 2 made it through the winter, 1 died of natural causes. Then we found another 3 this past year just at random. All of those lived until the transmitters fell off. over about one thing we had about 90 days worth of data on. And again, they're basically little miniature adults. They do everything that adults do at the same time. They just do it not at the same scale because they're smaller. So interesting findings so far. First, this is my old colleague, Brittany. She's taken a different job and just doing data entry basically now. But when we first found out that they're hibernating underneath the interstates, that was pretty interesting. So that the mortality rate, this grassy strip, and the creation of this new prairie habitat behind the admin building. Pheromones, neonates did not seem to follow their mom at all, which I would not have guessed. And then the sex attractive pheromone, whether it's terrestrial or volatile, has been a big part of my research. Seasonal movements, when they want to move ingress to a hibernation site in the fall, it is really, really quick. Again, maximum daily movement during that time, 328 meters. Egress, when they come out of hibernation in the spring and move away, much more gradual, takes a long time. Mating season, during the mating season, male snakes moving all over. So it really matters the time of year and the sex of the snake as to the movements. Site fidelity, this is one I wouldn't have guessed on, but basically the same, we had one snake hibernate in the same burrow 3 years in a row, one hibernate in the same burrow, two hibernate in the same burrow, 3 hibernate in the same burrow 2 years in a row. But not only that, but the snakes will come back to the exact same spots that they have used before during the course of the activity season. There seems to be some spots I think are just important, if you will, landmarks, if you believe it or not, for snakes, where multiple snakes will use the exact same log. And it doesn't seem to be a mating season thing. It can be multiple males that make it there. But I really think as they're moving through, I think snakes have some type of map. And Bob Aldridge would always say, snakes know where they are, they know where they're going, and they know what day it is. And I completely believe that to be true. Animals have a lot more going on than we ever give them credit for. Overwintering, right? Their thermal profiles, when they start coming up out of hibernation, I think will be really interesting to relay. Their movements, their distance to the forest edge, to trail, to people interactions. Surprisingly, I mean, it's not necessarily a random subset of people that are out at Powder Valley. It's people who want to be out hiking. So they're like, oh, that's a cool study. You know, that's really interesting because I think it's people who are already interested in nature. This seems to have a lot of support with the public. Again, it could just be that subset of nature lovers that are out there anyway, and maybe random people may not find it as interesting. But public perception and public awareness, I think, will be a really important part as we start wrapping up the study, which this year, 2022, is going to be the last year of that project, and then we're walking away. And as this is a nature group, you never know what you're going to find when you're out there. So I found this one day. I saw this hawk, this red-tailed hawk, on my way out to track. And he was just sitting, low-lying branch, just looking around. And I went out, half hour, 45 minutes later, I come walking back, and he's on the ground acting all strange. So I hide behind a tree and then start taking pictures. And it wasn't until later that I actually saw what was happening. But this hawk spent at least 20 minutes trying to get this chipmunk to come out from this hollow log that he barely scampered under. Or a squirrel, if there's a mammologist, I don't know. I can't tell. Some big rodent underneath this hollow log. But the hawk would jump down and he would poke his head in on this side. Nothing would happen. Then he'd jump down, he'd walk to the other side, he'd poke his head in, he would jump on the log. This hawk did everything to get that rodent to come out or explore every opportunity to get into it, and nothing happened. Like that little rodent never ever came out of there. Eventually the hawk gave up and flew off. But again, that was after 20 minutes or so. So you never know. Every day you go out in the field, you learn something, whether you realize it then or not. And if you keep your eyes open, you see all sorts of fun stuff. So I'll miss this project when it's over, but there's other interesting projects that I'm involved in that I want to start moving into. So anyway, I think that's about it. I want to thank you all for your time, for inviting me to chat. And if anybody else has any other questions, I'm happy to do my best to answer them. if you do.
I'd be interested to hear about their prey and any differences between what the babies eat and the adults.
Yeah, so we don't have any specific data on that, but I can talk a little bit in general about pit vipers and snakes. This is the second time that I've done a project with neonate snakes and compared it to adult snakes. The Massasauga was the other one, but snakes are gape-limited predators. And they have no hands, right? No front appendages, no rear appendages. So to me, it's always interesting how they eat or what they eat and how they eat it. They can't grab something and then turn it around and manipulate it to swallow it. And they have to swallow something, or they should swallow something head first. That way it helps press down the appendages and it's not all flailing around, right? It fits as a better packet if the appendages are all facing down. Basically what would be the anatomical term. Adduction. They wanted to adduct all the limbs and make them as close to the midline of the body as possible. But gape limited predators, they think that allows them to specialize on different prey items if the individuals of the same species are different sizes. So in a lot of species that have male-male combat, males grow larger than females, and a big male copperhead may be able to eat a chipmunk, whereas even the biggest female copperhead probably not. And the babies, you read everything from insects to worms to small rodents that they specialize on. But what I always come back to is particularly for copperheads, they're pit vipers. And pit vipers, it's a heat sensing pit. So they would have a tough time locating another snake to eat it. It's not going to show up on their thermal profile. A lizard, they'd have a tough time recognizing that except by vision and by scent. Their pits are adapted for warm-blooded animals. So mammals, birds, essentially. And if you're a snake on the ground, one that's not really even arboreal, you got to figure you're not going to come into contact with too many birds. I know there's ornithologists out there. So of course you have the oven bird, you have other ground nesting forest birds, right, that they could come across, typically not eggs. But my guess is both the adults and the neonates eat little rodents, mammals, shrews, voles. Shrews actually make a good meal because their head is not as domed, their skull's not as domed as the voles and mice. But pretty much any mammal that they can fit in their mouth is my guess what their diet's going to be. And the neonates just can't have that big of a variety because their mouths are a lot smaller than what the adult mouths are. So that'd be my best guess for that.
Ben, there is a couple of questions on the chat.
Okay.
First one was from Bert Knoll. I think it was Bert Knoll. Anyway, he's asking, what do you think the population density and how many is at Powder Valley, and how many snakes do you think are out there?
Oh man, I have the perfect answer for that, as I've been asked this a number of times. I am 100% confident the population size of copperheads at Powder Valley is between 1 and 10 million. Somewhere in that range is going to be the actual answer. How many there are, I have no actual idea. I thought when I started, it would be a pretty robust population. But the fact that, again, I should go way back. And one thing that really started having me question what the population size at Powder Valley was Captain Kirk. And this particular slide, right? Captain Kirk, within a 10-day period, basically covered, let's just say 15 of these 17 hectares looking for a female. He went every boundary, right? Until he hit a road or something, then he came back and went a different direction. And he stopped when he found a female. So he basically scoured this entire area for 10 days to find one female. I never found a male snake with more than one female throughout the entire mating season in any of the years that I was there. So in all honesty, I don't think the population size of Powder Valley is nearly as big as I would have guessed. I don't feel comfortable putting a number on it, specifically since it's being recorded. And I know how my students would be like, yeah, one time you said this, and therefore I'm right forever. You know, that you were wrong because you said this one time, and we have proof that you said it. So I really don't know. The fact that there's still babies and young snakes out there is a good sign, but we really don't find a lot of big snakes. And again, the bigger the snake, I think the easier it is to be seen. And there's a lot of things out there that will eat them. It's a very fragmented landscape, lots of edges. Predators like to work the edges. And I know that copperheads are predators, but they're very mid-level food web predator. The coyotes, the foxes, the hawks, they're all working those edges too. So I really don't know, but I'd at least say there's probably 100 to 200 to 500. I really don't know how many are out there at any particular time. And I'd say the rest holds true for any other species that's at Powder Valley. It's great for migratory birds passing through, but it's very fragmented. And with us, even though it's a relatively large habitat, 112 acres, it's small compared to what they once would have had in the Ozark Hickory Forest, Oak Hickory Ozark Forest here in Missouri and Illinois. So I don't know. I think that population is smaller than what we think it is.
Okay. Second question, somewhat related to the first one. James Falpall was looking at iNaturalist observations of the eastern copperhead, basically in our region, and noted that the closest urban population of copperheads has been reported in Powder Valley. Do you know of any other populations that might be also in St. Louis urban area? St. Louis County, St. Louis. Let's say he's wanting to know if you're closer. So obviously, I've seen copperheads out at Rockwoods Reservation years and years ago, but I think
Yeah,
he's
they have
thinking more in terms.
timber rattlesnakes there too. Yeah, I mean, I don't know for sure. There's two things I would say to that. One, they're very secretive, but two, people always know where there's venomous snakes. If you have venomous snakes on your ground, you've seen them, you know that they're there. I don't know, in terms of a robust population. Like I said, my old PhD advisor's son has had 4 in his crawl space just this past winter. So they're around, but I think wherever they are, people know where they are. So if the report says it's the closest, I'd be hard pressed to say that there's one closer than that. They need an expanse of habitat. I always look, I live in Kirkwood, and I always look around when I'm driving. I'm like, wonder if they could live there? And then I always think, I bet they did at one point in time. There's simply not enough chunks of habitat in a lot of these places where copperheads would still be able to hang on with a decent, viable population size. And usually for that, you think minimum 500 adult individuals to start avoiding inbreeding, depression, and things like that. So I really don't know if there's any closer, but things like iNaturalist are wonderful to help answer those questions. And as more data comes in over the years, keep checking. Maybe somebody will have one that's closer. I'm not sure.
We got an email from the Kirkwood School District a few years back that somebody spotted one on a walkway near Knifer, which is near the Magic House, for any of you who need a reference point. And people were going kind of a little overboard on worrying about it. But so there's some a bit closer.
Yeah. And with that, hopefully there was a photograph or something associated with it. A lot of times snakes get misidentified as another snake.
That's possible.
Yeah, that happens. That happens a lot of times. And even rattlesnakes. There's so many snakes that vibrate their tail when threatened. Racers do it. Water snakes do it. Garter snakes vibrate their tail, and of course there's no rattle, but if that snake is in dried leaves and sticks, it sure sounds like a rattle. People are like, it was vibrating his tail, I heard a rattle sound, and they're absolutely 100% confident that it was a rattlesnake. and it's a king snake or a racer or a rat snake. And then every snake in the water apparently is a cottonmouth. And I'm like, there's so many species of water snakes. Almost all of those that are reported as cottonmouths, usually, not all of them, but a lot of them turn out to be water snakes. So it does happen. There are certainly some instances of a snake, maybe it was in captivity and it escaped. Or maybe, just somehow wandered that far. Like, wasn't it this past year, there was that bear wandering around Webster Groves and Kirkwood,
Yeah.
that black bear that wandered down. And then eventually they tranked it and moved it off. So, I mean, it happens. You get some vagabond, particularly males, that are out moving around and then they get lost and they just keep motoring until they try to find some habitat. So, I don't know. Could be.
Okay, follow up to that question and I think you answered it early on but just if you could reiterate here, has anybody bitten by a copperhead at Powder Valley?
Well, besides me?
Have you? I hope you haven't.
Yeah, I have, but not since the project started. But like I said, before it started, there were 2 kids, different years, different kids that were shooing a copperhead. They didn't know it was a copperhead snake, trying to shoo it off the trail and got bit in flip-flops. But with snake bite, a venomous snake bite, people always want to hear about that, and that's a story for a different time. But a lot of times it involves hands. It's somebody picking up the snake, which they shouldn't be doing. I usually have a slide in here that, what to do if you encounter a copperhead. And then I have the Beatles. It says, let it be, but yeah, if you see a venomous snake, just let it be, walk around, move around it. I guarantee you they're more afraid of you than you are of it, no matter how afraid of it you might be. But most venomous snake bites occur because somebody's picking the snake up, and most of them are males, and those that seek treatment, over 70% of them have alcohol in their system. So y'all can make your own conclusions, or is it correlation is not causation type of thing, but you can make your own conclusions. Maybe inhibitions are lowered or who knows what. So
okay. Chris Brown had a comment. He had a copperhead that predated a wood thrush fledgling. Apparently he was radio tracking it, so he was doing some work just like you.
On the radio tracking the wood thrushes.
Chris, would you like to comment a little more on that?
Yeah, no, that's right, Ben. We were tracking wood thrush fledglings, and one of them ended up staying put for a little while under a log, and ended up finding that it was a copperhead.
Oh wow! So did it actually eat the wood thrush?
It had the radio in its system, and then later pooped it out. So yeah.
All right. Yeah, you never know what you're going to find.
Yeah, it's pretty cool.
You know what? I'm sorry. I'm definitely sorry that happened. But yay! You don't know how many times birds have eaten my snake. So we're on the scoreboard.
Here you go.
Yeah.
I was wondering when you were talking about the prey earlier, Ben, if maybe sun-warmed basking lizards might be, especially for the young, those baby Scalopris or something might be something they could eat. But if you haven't seen it, what can we say?
Yeah, I mean, I'm sure it happens occasionally. It might be, the exception to the rule. And in fact, we did a prey study with the innate Massasaugas, and we're like, all right, what is there a lot of in August, September at Carlisle, when they were born? There were shrews, there were voles, there were mice, there were grasshoppers. We got like little ringneck snakes, little brown snakes. We got lizards. And it was almost like a comedy routine when we put the lizard in with the baby Massasauga. The lizard kept walking around in a circle trying to get out of the tank, and the Massasauga just kept literally following it around. It was like it could smell it, but it couldn't see it, and it couldn't sense it with its pits. It's another pit viper. And it was literally just like, it was just like one was following the other, like a parade, just keep going around in circles. And it was, almost like the blind leading the blind. So I think they really have a difficult time finding those. They don't specialize on it as pit vipers, but a meal's a meal. And if there's happens to be an occasion where they come across one another, then so be it. We also had one baby Massasauga that was actually asphyxiated by what it ate, but what it ate had maggots on it. So we think it actually scavenged the shrew, because why else? We couldn't figure out any other reason why there would have been maggots on the shrew, except it was dead and starting the decomposition process. So I don't know. But it's certainly possible. As long as I've been doing field research for 23 years, you could tell me that bison migrated across the plains and hot air balloons. They'd be like, I can see that. Nothing is beyond possibility to me anymore. Like you see some crazy things out there sometimes.
Ben?
Yeah.
Ben, I've captured a number of non-venomous snakes over the year, and several times I've been squirted with that exuviate from their anus that was really smelly and disgusting, and I thought it was poop. And I read somewhere that it comes from what they call a repugnatory gland.
Yeah, musk gland. Yeah, they keep them down. The musk glands are, like you said, right near the cloaca, and they can squirt it and they can aim it. Believe it or not, they almost always hit you in the face if you have them picked up and they're measuring them, weighing them. And some of the musk from some of the snakes smells worse than others. To me, the nature scene snakes is awful. Garter snakes have a pretty bad musk. Water snakes have a horrible musk. Queen snakes, line snakes are even worse than that. So some people say copperhead musk smells like apricots. I never got that. I don't know, but as far as that goes, I think I'd rather be squirted with something with apricots scent than some of that musk. But you're right, some of it you can wash your hands and it stays on you for a couple days before it finally goes away. Your dogs won't even come around. It smells so bad.
Luckily, I've only gotten it on the hands.
Yeah, I have experience with that musk as well. Is there anything known to get rid of that smell?
Time. Time is the only thing that you can wash, and it really helps it go. It helps it dissipate, but you can smell. It's like, it's still there. And my old master's advisor, Chris, he loves smelling the worst things. He'd catch something and it would musk all over his hand. He'd go, "Oh God." Oh, it's like, "Come here, you got to smell this." And everybody would be like, "Get away from me, man." So there's not much you can do. You can kind of, like I said, use soap and water. That helps a lot. I'm sure maybe you can, what they do for dogs that are sprayed with skunks, you get the hydrogen peroxide, baking soda, and it's supposed to eliminate the odor, not cover it up like the tomato soup, whatever people would say. You make a concoction of baking soda and peroxide and what have you, and it's supposed to eliminate the odor. Maybe something like that would work. I've never tried that. I just deal with it because the next day I'm going to get more of it anyway. So
Yeah. We've got another question here from Miguel. He wants to know more about territories. In particular, do males have a bigger territory than females? And do males actually protect their territories?
Yeah, so that's the key is, you hear these different terms and, people love words and all the words have meanings and different words have slightly different meanings. So when you start talking about a territory is something that is actively defended. It's got some resource in it that's defended from other individuals of the same species. And it's something that they typically don't go out of. And there's individual set territories. No snake species that I know of has an actual territory. So what we call it instead is an activity range. Wherever the snake moves, right, that's its range. The only time snakes have ever been observed fighting is over lady snakes, is what it is. The males, they used to call it the snake dance, right? Where like they think that the male and female were dancing before mating. It's really two males fighting because there's a female nearby. So that's the only time where there'll be any confrontation. In fact, that's really the only time snakes come together is during the mating season for mating. They're all solitary. They don't live in groups. There's no packs. There's no herds or flocks or schools or gaggles or what is it, a murder for crows? I forget what eagles has a funny one too. I don't remember off the top of my head, but I'll think of it. Not a quorum. I'll think of it sometime. Anyway, but they don't come together socially for any other reason except mating. And those that communally den together, they'll come together then. But besides that, there's no territory. There's nothing that's defended. And the only time there's any physical interaction was for mating or for basically fighting for mating. So, which makes the spread of things like snake fungal disease, which is a different project I'm working on, really interesting. How does that spread if the individuals aren't coming in physical contact with one another? Hardly ever. So no true territories, but activity range, they overlap. Males move in, males move out, females move in, females move out. No real no real activity there, except they just happen to be in the same spot at the same time, just because they like the temperature, they like the sunshine, what have you. So.
I hadn't heard about the snake fungus disease. It sounds like another talk in the future.
Oh yeah, it could be. We don't have too much data on it yet, but we're working with the zoo on that project. I've got another project that Bob Aldridge and we've been trying to answer. We finally got really interesting data the last couple of years, so much so that, like I said, I've written it up. It's been reviewed. I just need to actually submit it. But all about pheromones and how snakes locate one another, whether it's terrestrial signals or volatile signals or both. And then looking at the whole history of snake pheromone studies from 1937, when the first ones were published, through present day. So that's pretty interesting. And I don't know, like we actually applied last year for your Bo Kester grant to expand some of the Copperhead work, but we weren't lucky enough to be selected. So maybe we'll try again this year and see what we can do. But Copperheads are winding up. Maybe we'll try to do it for the pheromone study and see if we can make it through your selection process this year.
Send it in.
Please
Oh
try.
yeah, we
Yes,
will. I
absolutely.
look forward to seeing the announcement. I'll tell my students to write it up. Like, okay, let's do it. Let's see what happens.
You got the odds are pretty good on that one.
Oh, all right.
They're not a guarantee, of course, but the odds are good.
Yeah. Well, I'm sure you get a lot of really interesting proposals, and I would not want to be on the committee to sift through and try to figure out one from another one and be like, rate the merits of one versus the merits of another one. But I'm sure they're all equally interesting.
One year we got 3 different studies on chimpanzees over in Africa, and we thought that was a little too far away, far afield.
Yeah, Webster Groves and Africa.
Yeah.
But hey, maybe you all get to take a field trip and go visit.
Although we did have another lady who we selected. She was doing something in Africa, and the original country she was going to had a civil war, so she went to another nearby country to do her research.
Oh wow.
So the Massasaugas, the research on them sounds pretty interesting. They don't seem to be too common from what I can tell. Do you have any like areas or locations that you would like suggest going to see them?
Oh, I'm honestly sort of bound to secrecy on this. And you can look back at some of my papers though, and the biggest population in Illinois is at Carlisle Lake, around the southern border of the lake. But during the spring and fall, when the snakes are in their hibernation areas, is literally patrolled. And if they see anybody out in the fields, they stop them and ask them to leave. It's a highly, highly endangered species. There's a couple populations in Missouri. Rich Siegel studied them for quite some time. Jeff Brigler, the state herpetologist, refuses to let any information out about his best sites about Massasaugas to the public. So I'm afraid I'm not allowed to tell you where the best sites are. But if you read the primary literature, you can get an idea of where they are. And I can't say any more than that. So I'd love to help you out, and I know people love to go out and take pictures and cross it off a life list, but I think I'd have my herpetology card taken away if I gave away specifics. So,
well, I understand. How's their temperament?
oh, the pit vipers are wonderful. They're ambush predators. They typically rely on camouflage, and I always tell students when we're out in the field, I'm like, if you see a snake, you hear a snake is moving away from you, the odds are it is a rat snake. It is anything other than a venomous snake. Venomous snakes just sit there. They stay camouflaged. Even when you pick them up, if you pick up a water snake, you're guaranteed to get bit like 6 times. And if you want to see something funny that you haven't seen, when you're done with this, go on the internet and search dirty jobs Lake Erie water snake. There's like a 2-minute segment of Mike Rowe getting bit over and over by water snakes. And it is true, the non-venomous snakes, they're really aggressive. When you pick them up, even before you pick them up, they posture, they do all these things. The venomous snakes aren't like that at all. Of course, you got to be extremely careful, use special tools and all that. But the temperament of the venomous snakes is always much more relaxed and chill, in my opinion, than any of the non-venomous snakes that I've ever worked with.
I agree with you, although seeing a fertile ants in the field, I'm going to have to disagree about that one.
Yeah. Oh, yeah. It breaks down out of the United States. You get the mambas. The mambas are like the velociraptors. They're extremely intelligent and aggressive. So you get stuff like that. And but in the United States, it's a little bit different. So.
Thank you.
Yeah. Thank you. Great questions, everybody.
So I did want to ask, after looking at your extensive list of publications, you obviously did a lot on the Massasauga, and that sounds like some really interesting research there. But knowing one of the species has got quite a reputation, and that's the brown snake that's in Guam, and you've got a publication on that. What exactly did you do for that? I didn't get a chance to read your publication, so I'm playing. But tell us a little bit about that.
I've never published yet on the brown tree snake on Guam. I've written grant proposals for study.
Maybe that's what I read.
Review. Yeah, it's in review. But basically, the whole idea of the pheromone project that I alluded to, that Bob and I are doing, and we started with the northern water snake, and we want to expand it this year to the red-sided garter snake, particularly, and then we'll see what other snakes we can get in decent numbers to do this with. But everybody has been convinced that the snake sex attraction pheromone, how the males find the females, is a terrestrial only pheromone, where the female crawls on the ground and deposits this pheromone trail that then males stick out their tongue and then follow the trail to find the female. But there's so many snakes that live in so many different types of habitats. There's arboreal snakes, there's sea snakes, there's semi-aquatic snakes, there's fossorial snakes. That would break down the medium of the habitat that some of these snakes live in is simply not conducive for leaving a terrestrial continuous trail. So we figure some of these snakes have to have a volatile trail, and some of those would be arboreal, or aquatic or semi-aquatic. So we started looking at the water snakes, since it's a semi-aquatic species, and we found this amazing evidence that the pheromone or pheromones that they use, they certainly can use terrestrial pheromones, which we were surprised to find, but they also use volatile pheromones. So our idea was to show that snakes use volatile pheromones. And then we have collected this particular pheromone in an organic solvent of hexane. We've had it run by chemists, and we are in the process of identifying the specific molecular compound of the volatile pheromone, and then synthesizing it in the lab, and then using it to attract males. And you say, well, that's all cool in theory, but who cares about bringing in male water snakes? And you're absolutely right. Nobody cares about that. Not even I care about that, and I'm studying it. The idea is to show a proof of concept that we can extract the pheromone, synthesize it, build it, and then release it in a volatile fashion to bring in males of a particular species, just like the Japanese beetle traps that people have in their yard. But then to use it for invasive species like the brown tree snake on Guam. So the thing that I'm thinking that you saw was this grant proposal that's out there for subgroup for the USDA to ask for funds, a large amount of funds to do the same thing for the brown tree snake, to travel to Guam, or at least to the couple places like in Colorado that have a collection of brown tree snakes, swab them, get the pheromone off it, make sure we have the pheromone by bioassays in the lab, then figure out what that pheromone is, molecularly synthesize it, concentrate it, and then use it to lure in male brown tree snakes to eradicate them from the population. We also looked to hopefully do the same thing with the Burmese python population, the Everglades, tegus, anything that would locate mates with pheromones. We're basically looking to build pheromone traps in order to make that happen, and trying to show proof of concept with a local native species first, and then showing that, hey, this works. Now let's step it up and use it for something that may do some good conservation wise.
Okay.
So you started talking about chemistry. I have to ask, what's known about the snake pheromones in terms of their chemical structures? Are they single compounds, multiple compounds? What kind of molecular weights? What kind of volatility at typical ambient temperatures?
Yeah, not a lot's known. It's a chain of saturated and monosaturated compounds, lipid-based, typically viewed as really heavy and non-volatile. You can look up, what's his name? Bob Mason had a paper in either Nature or Science in like 1989, where he lists the one snake sex attracted pheromone that's been found. The study's never really been replicated. They found something similar in other species, but it fails to elicit a response from males. So really not a lot is known. We're looking for something that we think is a little bit smaller, more lightweight, as it wouldn't necessarily be a terrestrial pheromone. I can't help you out much more than that, as we're still in the process of figuring out what this compound is ourselves. Like I said, there's one paper though that you can go and it gives you quite a bit of information. Rocky Parker is another person who's done some work on this. He was one of Bob Mason's students. His results are, to me, pretty inconclusive, and he and I just don't agree on a couple things, but that's the way it goes. So I can't tell you much more about the compound than what I did though.
All right, that's good. Early days then. Thanks.
Yeah, bottom floor.
Hey, anybody have any more questions? This was wonderful.
I have another question.
Okay, Pete.
It's about the specificity of antivenoms and different snake species. Is it pretty well individualized, or is there still a lot of research that needs to be done?
Well, I'm sure there's a lot more research that could be done. Is it itself has changed over the years, but typically what they do, and one thing that's interesting, if you take a tour, behind the scenes tour of the Herp House at the zoo, is on the back of every venomous snake's cage, they have in a big red sign with a white letter on it, A, B, or C. And it's basically based upon what type of viper is in there, what type of venomous snake that's in there, viper, pit viper, or lapid, what have you, what continent that snake is found on, and etc. As there are basically 3 main different varieties of anti-venom. So of course you'd want it to be specific to whatever species you were bit by. What they do for all the North American snakes is they take, it's 5, I think. I think they take copperheads, western diamondbacks, western diamondbacks, the Crotalus viridis, the prairie rattlesnake, and then 2 others. I forget what the other 2 are, but they take the venom from these 5 snakes. What they used to do is they would inject it into horses and let horses, small amounts into horses, and let the horses make the antibodies against it. But it turns out, and then they harvest that, of course, from the horses, and they sell it at a high price. But more people were dying from anaphylactic shock from the horse serum than they were from the effects of the bites itself. So they started, instead of using it with sheep serum, they started injecting it into sheep's instead, and that's made the process a little more commensurate. But they retail for about maybe $5,000 a vial. And if you're bit, you get anywhere, but depending upon the severity and your reaction to it, you get anywhere from 5 to, let's say, 20 vials of anti-venom. But like I said, for the United States, they take the 5 most common snakes, and the copperhead itself makes up 50% of all the reported venomous snake bites in the United States. Again, it's pretty wide ranging. It's secretive. It's found near people. For whatever reason, they make up 50%. They take all of those, inject them in, and then the venom itself varies from species to species. And it's a lot of big digestive enzymes. You get a lot of proteases, you get a lot of lipases that are in there. It's really for feeding. It's not for protection at all. And the venom itself has 2 main components, and varying species to species, these components can be in different propensities. You have a hemotoxin, and the hemotoxin, heme, blood, right? The hemotoxin ruptures all of your red blood cells. So the hemoglobin comes out, you have hemoglobin, your respiratory pigment, you're not getting oxygen delivered to any of your tissues, so you're no longer able to go through aerobic respiration and make ATP molecules very efficiently. So it's pretty nasty, right? You bleed out internally. It's like having a big bruise that never heals. So it ruptures as many red blood cells as you can. And then the other component of it is a neurotoxin. And a neurotoxin, right, is going to disrupt your neuronal signaling, either between 2 neurons, so a signal going to the central nervous system, or from the central nervous system to a muscle. And that muscle could be the leg muscles, so you could run away. It could be your diaphragm, so your lungs could contract. Your heart is a really big muscle that we have. But between the nerve itself and the effector, right, the muscle, the organ, the gland, what have you, there's a tiny little gap called the synapse. And the neurotransmitter, the end of the neuron, releases a neurotransmitter to go across this cleft. The neurotoxin stops that signal at that cleft, stops the signal from the neuron going either to the next neuron or to that effector. So meaning your diaphragm never gets the signal to contract. Your heart never gets the signal to contract. Your leg muscles never get the signal to contract and run away. So it's really nasty stuff. It's designed to paralyze their prey, make the prey bleed out internally as quickly as possible, and then to literally start digesting the prey as the snake is ingesting it. So it's pretty nasty stuff. I've seen a venomous snake bite a mouse. The mouse jumps up in the air, lands on its back, and is dead within a matter of less than a second. And I've seen it take a lot longer to work. Snakes can choose whether or not to inject venom when they bite. They can choose to inject venom from one fang, but not the other fang, or what have you. So again, it's primarily for feeding, not for defense, but they will certainly use it for defense. But all that venom then goes in again to make the antibodies that then get injected into you if you're unfortunate enough to have that experience and go through venomous snake bite.
That's great. Thank you, Ben.
Yeah, great question. People always want to know about venomous snake bite. Always.
Yeah, I guess it's after 9 o'clock. We still got over half the people that were signed on. So good job. Great presentation. I learned a lot. And as a thank you, we are going to give you, Ben, your membership to Webster Groves Nature Studies Society.
Oh, cool. That beats my $10 a day I got for jury duty. This is awesome.
Good.
So this is the end, and I hope everybody has good holidays. Stay safe.
Yep.
Stay well.
Yeah. Thank you all so much.
Wonderful.
Thank you very much for coming, Ben. I really appreciate
You're
it.
welcome. Yeah, no problem. Thanks for the invitation, and thanks for having it go so smooth, Rich. You gave me everything I needed ahead of time and told me how it would all work. So thank you so much.
Did a good job. Did a real good job. Look for
thanks.
it probably won't make it to the January newsletter. It might make it into the February newsletter, your review.
Oh, okay.
I'll
Yeah.
send you a copy of it when
My
it gets there.
scathing review.